think we're heading straight for our very first speaker. We start with a fantastic presentation on a topic that I find extremely exciting. And I'm delighted to introduce you to Dr. Pascal Chavat from the INRA, the French Institute for Agricultural Research. Pascal started her career here in Newmarket, working with Peter Rossdale just a few years ago. and from here, returned to France and has been working with l'INRA and is with across various species, not just the horse, not just equines, but has also developed a very special interest in the genetics, epigenetics of the horse from conception through to birth and beyond. And she's also working in that context, in research projects around assisted reproduction, which all come into the sphere of trying to and developing further insights into the developmental and reproductive physiology of horses and equine and other species. So Pascal, please, the floor is yours.
Like that? Does it work? Yeah. Well, thank you very much for this very nice introduction. I'm very proud and glad to be here. I don't work on thoroughbreds anymore. I work on more sports horses and sometimes ponies as models. And so I will talk to you about the effect of mare nutrition and also the management of the mare on the development of sports career of the offspring. So we'll first very shortly set the scene. I removed very complicated molecular slides and talk to you about a bit of epigenetics and critical period and the developmental origins of health and disease. Then we'll talk about work that first started here in Newmarket with Twinkellen and that we followed up, which really gave a proof of concept of this concept of the developmental origins. Then I will tell you what we know about its importance for your practice for horse breeding, including some data that has not been published yet, so not yet validated by a publication, but we're pretty safe on it anyway, and I want you to think about it, and then I'll finish with concluding remarks. So my whole lab works on the developmental origins of health and disease. And it all started with Dr. Barker from University of Southampton, who noticed that the birth weight of babies was the most important character to predict the onset at age 60 or more of death by cardiovascular disease or diabetes onset. And I first went to listen to him with Peter Rossdale, who was sitting next to me just right at the beginning. It must have been in 1989 or 1990. And Peter said to me, Pascal, if it works in humans, it works in horses. And it does. So we know now that adverse nutrition, whether it's overnutrition or undernutrition, or adverse metabolic status like insulin resistance of the parents and the male also, but I won't talk about the male, will actually induce adaptations, so I don't know how I point out, in the development of the fetus, the embryo and the fetus, and there will be adaptation in the transcription of genes, the growth of cells, the growth of tissue, the growth of organs, and the molecular biology behind that is epigenome. So epigenome is, you don't need to know all the mechanisms, it's what goes over the genes. If you have genes of your skin and your eye, of course you are an individual, so you have the same genes. However, these genes do not express themselves the same way because some are programmed to do skin and some are programmed to do eye. And the epigenome, it was what goes on the genes and changes the expression of these genes, even with the same genes. Okay? So these fetuses, they adapt. And that favors their immediate survival so that they will actually live, especially when they are in adverse conditions. However, these adaptations of the epigenome, most of them stay, not all of them, but they actually remain. And so they remain as an adaptation to the environment that the fetus encountered in utero. So if you've been restricted in utero, you're adapted to an environment in utero postnatally, which is usually not what happens. And then, so we talk about this predictive adaptive response. Actually, this phenomenon now is known to play a role also in evolution, very short, rapid evolution compared to Darwinian genetic evolution. And so this fetus is not necessarily adapted to the postnatal environment. However, because biology is biology, it adapts. And it seems very sound. However, with age, with excess nutrition, aging, then these defenses, this mechanism of adaptation, they start not to be as good. And then you have an onset of metabolic disease, type 2 diabetes. It's been shown also, I put osteoporosis because in humans it's also been shown to play a role in osteoporosis, obesity, and many, many other diseases. Even mental diseases now are known in humans to have some root also in fetal life. So initially, when Barker talked about that, we had no idea about the mechanisms. 1990, nobody knew. Now it's been shown, and it's the object of the DOHAD Society, International Society for Developmental Origins of Health and Disease. And of course, like Peter said, if it works in horses, it works in other species. And so we'll focus on the horse. So the developmental origins of health and disease, it's quite logical, because not only will the recipient or the pregnant mare host the embryo, she will carry the foal for nearly 11 months and nurture it afterwards with her milk. So of course she plays a role in the development of this foal. So the proof of concept is based on the fact that in the horse you have horses of different size, as you know, and that determines their uterine capacity. And in the horse, you have only one offspring, and the placenta covers the whole uterus. So if you play with horses of different sizes, you actually will restrict or increase the size of the placenta. And because the fetus is fed by the placenta, you may be playing on the growth of the foals. And what Barker had shown already, I told you, it's the baby that were small that were actually developing the disease in late gestation. So in horse, we can enhance fetal growth or restrict fetal growth beyond the genetic potential of the animals by varying maternal size. So there are many experiments that were first started with Twink Allen, and then we revisited it because we wanted to know about metabolism. So that was the thesis of Pauline Penier. And what we did, so I actually mixed the results of Twink and ours so that you get a whole overview. So we collected embryos and we made pony controls. And then ponies were transferred in thoroughbred recipients. That was in Twinks. We used saddle horses, or we transferred them into draft recipients, trying to make what we called enhanced ponies, big ponies. We call them the sumos at the lab. And effectively, the sumos had larger placentas, and they were larger from birth. And as you see here, the control ponies, is there any pointer? No? So, well, you'll just have to see. So the yellow line is the normal growth of ponies, and the blue line is the growth of the enhanced ponies. And as you see, it goes over lactation, and they still are 30% larger at 540 days, which is almost 2 years. Not only are they larger, but their growth seems to be pretty harmonious. You see that all these graphs are more or less parallel. So the growth is parallel. So they seem to be very harmonious. However, that's the work done in Cambridge with Abby Fowden. When they actually analyzed these foals in very early stages, at 6 days of age, they had some differences in the arterial blood pressure and the heart rate in the ones that were enhanced compared to controls. So there were some adaptations already in the heart rate and blood pressure regulation that were disturbed, which of course in the long run might play a role in performance. So they have elevated blood pressure, they were less sensitive to drugs that can affect the blood pressure, and they did not respond very well to stimulation. For us, we looked at the glucose metabolism, glucose being really the energy of the cell. And you see when we actually injected glucose, the ponies were actually, ours were resistant to insulin. They had to, in blue, put much more insulin to actually keep their glycemia down. So they were resistant, which is not very good. When we did the test again at 200 age, and I don't go over all the tests that we did because they're a bit different, the golden exam that you do is a clamp. There was no difference at weaning, and there was no difference anymore. Also, although you see a wide variation at almost 2 years. And then we did the other way through. So after we had enhanced the growth, we tried to restrict the growth. So again, same system. We used thoroughbred embryos that was twink that he put in pony, and we used saddlebred embryos that we put in ponies. I didn't dare using draft mares in ponies. I thought I could go to catastrophe, so I didn't do it. And so in pink you have the restricted foals, and in green you have the normal animals, the controls. And again, they had read the books, so the placentas were smaller, associated with the restricted growth of the foals. So when you look at the growth, so we also had enhanced saddlebred here. We see that the enhanced animals, the restricted animals in pink, they actually grew less, also after weaning, minus 17%. However, what's interesting here is that, is that the curves are not that parallel. Here you see things happening on the cannon bone. If you're doing show jumping, the cannon bone is really important at the reception. So the cannon bone was smaller. You see that it's not as harmonious as the foals. And so the growth of the restricted foals is slowed until weaning, but there's a disharmonious catch-up growth. And that can be a problem, and it's shown also in humans to be a problem. So we might have more problems with the restricted compared to the enhanced. And then when we look again at the metabolism, so it's the contrary. We have a better insulin resistance here in the restricted animals, and then it goes on until weaning. They're more sensitive, as if it was a positive relation, and then this disappears at 2 years of age. What's interesting, however, is that in humans, when you see that the curve goes on, unfortunately with my experimental animal, I cannot keep them longer, but usually it goes like that, and then they become resistant, and I don't have that data. We looked at osteochondrosis. We didn't do it in ponies because ponies do not develop osteochondrosis, and we didn't, as you see, we didn't have a lot of animals, but all of them had of the restricted one, they had osteochondrosis at 6 months of age, and compared to the controls and the enhanced, which didn't seem to have an effect. And they still had a much larger incidence of 33% at 18 months of age compared to the others. So in summary, for this result, we showed that if you enhance growth or reduce growth, then you have long-term effects, at least until almost 2 years of age. In terms of the maternal environment, you have a difference in nutrient capacity. In the glycemia, ponies have a higher glycemia than horses, so the environment is slightly different. And of course, the milk production is different. So we have an increase or restricted fetal growth. We have large and small placentas. The enhanced pony don't seem to adapt. They seem to grow normally. Whereas, in fact, which I didn't show you, the restricted animals, they actually try, there are adaptation in terms of, especially this gene, IGF-2, which is a gene, a factor enhancing growth. So they are restricted, so they reduce the growth. And so they have a different catch-up after birth. The restricted horse have a tendency to be hyperglycemic as a basal. They have restricted non-esterified fatty acid concentration. They have less fat. They have early improved insulin sensitivity, and then it disappears. Everybody actually at 2 months of age seemed to be relatively normal in terms of metabolism, and we had increased osteochondrosis in the restricted horses, and also, again, similar to these growth hormones, we have an increase in thyroid hormone concentration in the restricted horses, as if they were trying to catch up, and restricted in the enhanced ones. So we've made the proof. It can be important already for the industry of embryo transfer. It doesn't concern thoroughbreds, but what now we advocate is that when people do embryo transfer, they should have recipient mares that are about the same size of the expected size of the embryo when it will be an adult, and that's really important. And we could also even play with it when you have ponies, which are really double ponies, and you want to stay in the pony category for competition. You could actually transfer the embryos in smaller mares to make sure that your pony will stay a pony and compete with ponies. You could actually play with it or make them bigger like that. So some people already think about it. Anyway, so what does it do for the importance of horse breeding? As I told you, when we played with that, we also played with the glycemia of the mare because the horses and ponies are different. And I also told you that in humans, it's the birth weight that is important, and that really plays a role on the glycemia of the foal. Or we know, by the way, we know that insulin resistance becomes a real problem in the horse, and so it may result from the nutrition of mares. We know that the nutrition of mares can influence laminitis, hyperlipidemia, and here we are going to focus on osteochondrosis and diabetes in the offspring. So what do we know about osteochondrosis? There's going to be a lot of talks about osteochondrosis today. We know that the genetics play a role, the breed and the genetic heritage of the animals, but we know also that the addition of cereals in the diet, excess cereals, and postprandial insulinemia plays a role in the development of osteochondrosis in the foals, and that the postnatal environment, whether it's biomedical trauma, mineral imbalance, growth rate, housing, exercise, and the nutrition and carbohydrate metabolism will play a role on OCD. And you see there's a huge list here, and I'm certainly missing some papers. So we first started to work on that in 2010 when I met somebody from the Center of Mont-le-Soir in Belgium, and the Center of Mont-le-Soir is specializing in all juvenile problems of joints. And I was with the boss, and he was saying, oh, I'm very excited. I'm going to do in all the Belgian area, look at the nutrition of the foals and try to see how it influences osteochondrosis. And I was like, well, please look at the nutrition of the mare, which he did. And in the end, we showed together, this is the first paper, that the nutrition of the mare, because there were lots of situations, so we actually narrowed it down to whether the mare was only fed on forage during pregnancy or whether she had concentrates. So we didn't have the nature of the concentrates, the quantity of concentrates, but there was a really big effect of the nutrition of the mare and the postprandial insulinemia. And I didn't put it here. And so the foals whose mares were fed on forage only at 8 times less incidence of OCD in their population. So their population were sports horses. The incidence is higher than it is in the thoroughbred, but still, it was totally striking. So then, as a researcher, I don't believe very much until I have actually a controlled experiment to actually show that it's true. So which we started. So we started to supplement horses with cereals. So in the pregnant mares, we checked that we had postprandial hyperinsulinemia and hyperglycemia. And pregnant mares naturally develop insulin resistance, but when they were fed with cereals, they develop more vascular insulin resistance. And when we examined the placenta, we actually saw signs of inflammation and also vascular alterations that were related to the fact that the mares were fed with cereals instead of just forage, and we had an increase in osteoarticular lesions, which was quite interesting. When we have undernutrition, we thought, well, maybe it plays a role also. We had effects on the foals with a delayed testicular maturity in foals that were castrated at 1 year of age. We had an altered metabolism of the pancreas, and also we had an altered growth, again, of the cannon bone. So even also in restricted environment, we didn't have any significant effect on the osteochondrosis. It would need to be done again because, again, this is research animals and we cannot afford having hundreds of animals. But we actually saw the effects of the nutrition and especially cereals being really at stake in that. So then we thought, well, some mares are fed just during pregnancy with cereals. But when you look at the global population, and there's a lot of epidemiological studies in horses in the UK, but also all over the world, showing that a lot of mares are overweight and especially obese. And we know that a lot of, especially the occasional breeders, they have no idea of what is a fit horse and what is the normal BCI of a mare, BMI of the mare, and so they have obesity. So we thought, what happens if the mare is obese at the time of pregnancy and then she's fed the way people feed horses? So this is not our work, this is work from the United States. So in early gestation, we know that in the endometrium, there's also inflammation, cytokines, which are inflammatory factors, alteration of the lipid regulation, and mitochondrial stress. And the mitochondria is the organelle in the cell that makes the energy. In the embryos, they are disturbed. They are more inflamed. They have altered lipid regulation, stress, oxidative mitochondrial stress, and they have an altered uterine environment at 8 and 16 days post-ovulation. So already, by the time the conception has taken place and this embryo is in this uterus, and in horses, the implantation is late. It's about 38 days. So this embryo is depending on uterine secretions to grow. The nutrition of this embryo is different. Then, we played again. So we had obese mares. They were mares. They were not ponies. And with obesity, as in humans, you have an increase in insulin resistance and inflammation. And that's in all species. So we see placental adaptation. What's really funny, I won't go into all the details, but we see the same type of placental adaptation as we can see in humans that have type 2 diabetes. So, and we didn't see an effect on foal growth. And that's what's important for you. You will not see, in those cases, effects on the foals. That's very deceiving because then they're sold and you don't know what goes on. However, at 6 months of age, they had increased SAA, low-grade inflammation, like the mares, increased insulin resistance at 6 and 18 months, and increased osteochondrosis at 12 months of age. Then we thought, well, maybe it's due to the milk production of the mare. So we just published that. We looked at the milk production, and the milk production was reduced in the mares that were obese. However, there seems to be compensatory effects because the growth of the foal was normal, but there was a modification of milk quality with increased fatty acid, which could play a role in this effect on growth. However, the composition of fatty acid is slightly different with increased pro-inflammatory fatty acids, so omega-6 compared to omega-3. All horses were, of course, in exactly standardized breeding conditions. Otherwise, if they are not fed the same, you cannot compare. So basically, both under and overnutrition of the mare can alter long-term offspring health in terms of metabolism and bone development. And then over 10 years of research with the same herd, we had primiparous mares and multiparous mares. So we always noticed, so when we came back to do all our statistics, all the foals from the primiparous mares were smaller statistically than the multiparous mares, which is known. There's a lot of papers, so what we have seen and others have described, so impaired metabolic adaptation. I told you the mares, when they're pregnant, they become insulin resistant, which means that there's more glucose available to the fetus, and it enables the fetus to grow. And it's a normal adaptation in all mammals. In women also, we become more insulin resistant during pregnancy. However, these changes, this adaptation did not take place as well or as deeply in the primiparous mares. They don't adapt as much, as if they needed to be trained. They also have a reduced placental exchange. So it's been shown by Twink, by a group in Brazil, and by us. So, we're pretty sure about that. In terms of lactation, so they produce smaller and lighter foals, and again, it comes from all over the world. Everybody actually confirms that primiparous mares produces smaller foals. Then, in lactation, in terms of milk production, it's really inconsistent. There's only 3 studies, and not everybody agrees. In terms of the growth of the foals from primiparous mares, we still have a reduced size and weight until adulthood. Again, lots of work. Also, the foals, their glucose metabolism adapts with growth, like in children, and this adaptation seems to be delayed. And we also found delayed testicular maturation. So we thought, well, that could be a very physiological way to look at very physiological growth retardation, because any mare will have a first foal, and what we went on with the study. One of the problems with that work is that you can have old primiparous mares, young primiparous mares, and when they're compared to multiparous mares, usually they're older, and so there was no paper actually deciphering the difference between the effect of age and parity, and we wanted to know, because we wanted to help breeders saying, well, is it okay if, especially in sports, your mare does show jumping or dressage until age 15, and she has her first foal at age 15, is that bad or is that good, or should you actually make sure that she has a pregnancy during her career when she's younger? So this was the thesis of Emily. She's not finished to publish everything, but now she's working on human placenta, so she's lost for me. I think human placenta is much more interesting. But we know that in terms of racing, one of our speakers showed that performance and winning prizes is possibly correlated to size at 1 year of age. And, that we just showed that parity could play a role in the size at 1 year of age. And in show jumping and racing, we also have the performance that is slightly reduced when the foals are born to primiparous mares. So the thesis of Emily was to see whether age was playing an individual role where Parity was playing an individual role, and because she was a hard worker, I added lactation. Should we breed mares every year, or is a concurrent lactation where the mare is producing milk at the time that you're breeding, is it good or not for the embryo and for the development? And because I put her quite a big challenge, she actually looked at the embryo, the placenta, the lactation, and the foals. So I will just give you the results. I could spend hours to show them. I will just summarize the results. So when we looked at the effect of Parity, we confirmed that the primiparous mares produce smaller foals than the multiparous males. When we looked at the placenta, we have an increased gestational length. So they are smaller, but there's still kind of a compensation. The gestations last longer as a mean. So normally the foals should be heavier, but they're still smaller in size. The placenta is smaller in size. The efficiency of the placenta, the capacity of the placenta to transfer nutrients to the fetus is reduced. There is a similar structure of the placenta. It's not altered. There's a slight alteration in gene expression, which deals with fetal-maternal communication and transfer of nutrients. So there is an adaptation of this placenta, which makes everything he can or she can to increase the birth size, but the birth size is still smaller. So Twink was talking about a priming of the uterus. He was saying that this uterus had to have a first trial to be totally efficient. That we don't know yet. And we looked at the embryos, right? So there were the 8 embryos, and we also saw a slight alteration in gene expression. So already by the time of the embryo, 8 days after fertilization, there is alteration, and in embryo-maternal communication, same type of factors as in the placenta. And there's papers in mice showing that the environment at the time of breeding is really important. And I'm sure it's going to be the same with the mare. And so the nutrition at the time when you send your mares to be bred is really important. And I'm always worried about what's the role of getting in a lorry, going to the stallion, coming back, having sometimes different feeding. It would be nice to be evaluated. And then these animals catch up, and at 18 months of age, which is always they were the same size. And when we looked at the lactation, we had a reduction in the milk production, but we had an increase in milk quality. So which actually could account for the catch-up of these primiparous that actually grew larger. And that may actually, however, play a role in long-term programming of the performance. Then we looked at the effect of age only. So we decided to put a threshold at age 10 because there are papers, I don't think I put them here, showing that the fertility of the mares, there's 2 very large papers, one from France and one from somewhere else, where they show that the threshold for the start of the reduction in fertility in the mare is about 10 years. So we decided not to go to very old mares, but just to say, at the time when fertility is slightly reducing, does it play a role? So in the embryos, we saw a higher alteration in gene expression in the mares that were older than 10, with a delayed or poor embryo development, which actually could be related to the fact that these mares are becoming less fertile. So the embryo is affected, and because they are poor, they are more delayed, it could actually account for very early embryo loss and the reduction in fertility. So then we looked at the placenta. So the placenta increases. There's an increase in gestational length. There's no difference in the size, but there's different structure, definitely in the older mares. And there's an alteration in gene expression. So we have not published that yet, which could be due to the fact that this uterus of older mares is, so they're all multiparous. They've all had foals. There's degenerative changes taking place. And so the placenta adapts to actually extract more nutrition for the fetus. And it's actually an adaptation to maintain optimal fetal growth. And these animals are the same at birth. In terms of lactation, we have no difference in milk production. However, there's an increase in proteins in the milk of the older mares. But there's no difference in long-term morphology. So basically, the aging of the mare seems to, at least not very old mares, seems to be, we have embryo adaptations. Also, the oocytes, there's other work, especially coming from the United States, showing that the oocyte might also be disturbed. And, but however, once the pregnancy is established, the placenta compensates and those foals do well. When we looked at the effects of nursing, we couldn't get the embryos. So, we had a problem. There was a heat stress. It was in the full summer. So we couldn't look at the embryos. So I don't have that data. However, in terms of placenta, when the mares were nursing at the time of breeding, and we actually bred all those mares not at the fall heat, but at the second heat. We decided, again, to standardize, because otherwise you don't know what is the effect of fall heat and not fall heat, so we had to standardize. So reduced gestational length, no difference in size and structure, again, alteration in gene expression with placental development, but here we see immunity and inflammation taking place. So we're wondering whether the placenta adapts because of changes in maternal nutrient availability. This mom has to actually give milk to her foal as well as developing a pregnancy. So she has to, there's a partition in the nutrition, nutrition repartition. However, at birth, we have no effect on the morphology in the foals that were born to nursing and non-nursing mares, which was very good to me because my stud manager at the experimental farm was always saying, oh, no, I need to breed every second year. And I was like, no, 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 no. So I was quite happy of these results. So we seem to have more efficient pregnancies when they're actually nursing. And, however, at weaning, the non-nursing, the non-nursing were actually smaller than the nursing, which was unexpected. And we had a decreased postnatal growth, but it was just before weaning, in the last, from 4 months of age, not at the beginning. And what we had is, we had a difference in the production in the long-term milk production in the quantity, but not in the quality. And we're just wondering whether we should see, or you should look at your foals and be ready to supplement as soon as you see a decrease at 4 months of age, those mares. So it is to be done again, because this was not big numbers, but basically it's really reassuring. However, keep an eye on the growth of those foals just before weaning. So now it's the last. So I told you about the nutrition of the mares and how it could influence insulin resistance. That's important. So we took that to breeders, and then they come to me and they say, yes, but I bought this mare, and she was fed concentrates all during gestation. What should I do to prevent the problems? And I'm like, well, I don't know. So we started to think of this insulinemia. Insulinemia is related to the onset of osteochondrosis, and very old papers by Jeffcott showed that repeated hyperinsulinemia in the foals increased the risk of OCD, but whereas when there were no concentrates from 6 to 18 months, and that's the Belgian groups that have been showing that in an epidemiological study, there's a regression of the most important lesion in some individuals. So we thought, there is something about that. And all those foals are usually fed twice a day with concentrates, at least in the winter. What do we know about the distribution of meals? When there's one, again, old paper here showing comparisons in the plasma glucose with feeding in animals that are fed 6 meals a day or 2 meals a day. And we see that there's a higher increase in glucose after the meal in the ones that are fed 2 times a day compared to 6 times a day. So we thought maybe there is something there. And then when we come back to our results, when we were looking at the mares that were fed forage or concentrates, you see in orange the ones that were fed with concentrates. So they were fed twice a day. And we see this large increase in glucose up after the meal, and then we're back to normal. And then the increase in glucose is not as high in the second meal, and we see the same with insulin, large increase in insulin, and well, less increase in the second meal, whereas the controls here are fed with forage. So we thought, well, maybe there's something to play. So Marion did her master's with me, and our farm is equipped with automated feeders. Automated concentrate feeders, like you see also in cattle, where you can actually, horses are equipped with a tag, and they can, it's like the automatic meal feeders also that you see in cattle, and so they come and they get only what the machine wants to give them. So we decided to have foals after weaning, so they were born in the spring, they are in pasture all summer, and at the beginning of the winter, they're put inside. They're usually in groups. And so you have these automated feeders that deliver exactly the same quantity and the same quality of concentrates. The only difference is either they had it in 2 meals a day, either in 8 meals a day. And I didn't tell you, but there's very often difference between males and females. So of course, all the statistics, everything is done separately between fillies and colts. So we had 8 meals with males and females, 2 meals with males and females, and before at the time just after weaning, we did the metabolic test and the X-rays, and we did it again when they went out of winter conditions. In terms of growth, there was no difference. So 2 meals is always in kind of yellow, and 8 meals is in blue. No difference in the foal weight. No difference in the average daily weight gain between the groups. If I don't put the sex, it's because there's no difference. However, in glucose challenge, as I told you, the response to glycemia changes with time. So we see very well the difference between 6 and 12 months, but there's no difference between the groups. And to cut a long story short, we have no effect on the numbers of individuals affected by OCD, no effect on lesions, the number of lesions. And then we thought, it's very difficult when you have a small number of animals to actually be very precise on OCD. You just have to say yes or no, and you cannot take the joint into consideration. So then we analyzed again a totally different group from France, analyzed the same X-rays that were taken by the Belgian group and analyzed them using the Denoise radiographic score, and again, we didn't find any difference. So basically, the increase in the number of meal size, at least on these parameters, did not make a difference. It can make a difference on ulcers or things like that, but that was not the object here, and so we were very disappointed by these results, but we will finally publish them. However, I have colleagues that looked at the intestinal flora and the metabolome, and there are differences. And so we are currently, again, the paper is in the process of being written, but the intestinal flora of the ones that were fed 2 times a day is more closer to that of diabetic humans compared to the ones that were fed 8 meals a day. So the story might not be totally finished here. So, and it definitely deserves more attention and to try and see how we can use these. So in conclusion, what I've showed you is there is an effect of prenatal environment, and I think they're totally underestimated. They must be estimated. The life of those foals starts from conception. Even the gamete production, the mares and the stallions, there's a lot of proofs in other species, even the stallions, so you also have to pay attention to the nutrition of the stallions, and there needs to be further evaluation. We're going on with that, but there's not many groups working on that, and anybody wanting to join the club is welcome. the choice and the nutrition of the dam are essential. And we need to have these markers of prenatal programming. Because at the moment when people, and we were discussing with one of the speakers this morning, when people come to me and tell me, what can I actually know? How can I know whether my mare is really at the level where she's really programming adversely my foal? Because most of the breeders, they have to give concentrates. And we've put the threshold at around 100 grams of starch per meal. But it's a bit empirical. There's one paper from Georg from Germany showing this threshold. But I cannot tell this person, unless we do really sophisticated tests, whether they have to fear of a problem or not. And we need to have quickly available, relatively cheap markers. And that's really urgently needed. I can tell you it has an effect. I cannot tell you to what level it has an effect and to what level you can be safe. So that's really important. And also, I showed you a lot of physiology, but I told you at the beginning that the mechanism is epigenetics. And at the moment, there's not a lot of money, there's not a lot of work in epigenetics. I just had my first grant on epigenetics in the horse. Because the funders, they want to have the results, but without understanding the real mechanisms. However, I think we will need to understand these mechanisms in order to actually find better solutions. So that's a plea for new generations to actually focus on that. Because that costs, at the moment, it costs a lot of money, but it will reduce. And so why is that happen? When I was at school, I learned that the phenotype results from the genetics and the environment, and there's an interaction between genetics and epigenetics. I don't know why it appears, but also the interaction. However, the epigenetics, so the environment, plays a role. And this is a paper that we published with my husband on the genetic performance of show jumping. And you see that the residual, so the genetic value for show jumping accounts for only 24%, 24.6% of the performance. The genetic maternal value, that's whether you change the recipient mare's 1.4. The permanent environment of the horse, 24%. That means that there's 49%, almost 50% that is not explained by postnatal environment nor genetics. Part of what I show is explaining this part of the residual. And there's really work to do, and in the prenatal environment. So there are the interactions, the genetics, the epigenetics, and the environment, the postnatal environment. However, I think there's another area where we need to focus is the metagenomics, so the flora. The gut flora is playing a role. It's known now for treatments, like for obesity, you can actually change the gut flora of people and actually reduce the obesity. There's work where you can have gut flora exchanges, and it really affects the phenotype. So we need to focus also on what is that doing to our foals and to their further performance. So I think I finish with more questions than responses, but I just want you to be aware of this is really front of science data. And of course, we're not getting all the answers all in one time. And with that, I want to thank all the people that have been helping me and funding all this work, which I summarized the 15 years of work in half an hour. And thank you.
Thank you very much, Pascal. I think that last formula you put up there, that's where it's all heading for, because I think we all grew up, or like you, with a very short formula, P equals, and two symbols, and now the symbols are gathering pace and importance too.